Assalamu alaikum, ladies and gentlemen. I welcome you all to Phenomenal Medico. Today I will talk about the fatty acid metabolism, and particularly in this video we will talk about the beta oxidation. So first of all, from the intestine where it is absorbed, the free fatty acid is then complex with the albumin in the bloodstream. This free fatty acid albumin complex is then transferred to the other tissues where with the help of this transmembrane protein it is co-transported along with sodium inside the cell. Once it get inside the cell, it is then complexed with the Z protein. This Z protein has a similar role in the fatty acid metabolism as the albumin had in the bloodstream. I mean, this Z protein get complex with the free fatty acid, and it remain in this complex form as long as the fatty acid is present inside the cytosol. Now, this free fatty acid in the plasma, its concentration is directly proportional to the uptake of free fatty acids by the tissues. So how this plasma free fatty acid concentration is maintained, it depends on many factors, like a few of them I have mentioned, like insulin and glucose. They have negative effect on the free fatty acid concentration, and the other are the catecholamines, the growth hormone, the thyroid hormone, glucocorticoids, the starvation process, and the diabetes mellitus. It is a pathological condition. They increase the plasma free fatty acid concentration. So how the insulin and glucose and other substances, they increase or decrease the plasma free fatty acid concentration? They involve, first we would talk about the insulin and glucose, that they involve interaction with this transmembrane protein. This transmembrane protein is linked to the adenyl cyclases enzyme. These adenyl cyclases enzyme are deactivated in response to the insulin and glucose interaction with this transmembrane protein. While on the other hand, when these substances like the growth hormone, thyroid hormone, the catecholamines and other substances, when they get attached to their respective transmembrane protein, this transmembrane protein in turn activate the adenyl cyclases enzyme, and the cyclic AMP concentration would be increased. This cyclic AMP in turn activates the protein kinases present inside the cell. These protein kinases further phosphorylate certain lipases enzyme in the cell. These lipases enzyme, once get phosphorylated, they become activated and they start breakdown of the cellular lipid, and hence the free fatty acid concentration would be increased. So this is how the insulin, glucose and other molecules, they increase or decrease the plasma free fatty acid concentration. They actually involve the interaction with the adenyl cyclase enzyme. So now this beta oxidation, it is a major pathway by which the fatty acids are degraded in the body, and it occurs in the mitochondria. The first step of the beta oxidation is the activation of this fatty acid. It gets converted to acyl coenzyme A by the addition of this coenzyme A, and this reaction involves the hydrolysis of this ATP to AMP and inorganic pyrophosphate. This inorganic pyrophosphate is then hydrolyzed further into two individual phosphates by the inorganic pyrophosphatase. So overall we can say that one acyl coenzyme formation or the activation of a single fatty acid molecule involve the hydrolysis of two phosphate bonds, one here and one here. So the hydrolysis of two phosphate bonds of a single ATP molecule. So it is an important point to be noted here. The enzyme required for this reaction is acyl coenzyme synthetase. It is also called as thiokinase enzyme. There are various types of the acyl coenzyme synthetases. I represented it on the right side of the board. The type 1 is mostly active on the shorter chain fatty acids like acetic acid, butyric acid, and the next one is mostly active on medium chain fatty acid like containing carbon number from C4 to C12. The next one for the long chain fatty acids, the C8 to C22, and there is a fourth type which is recently discovered, which is mitochondrial GTP specific, and it involves the hydrolysis of the GTP into GDP and inorganic phosphate. So this was the activation of the fatty acid, which is the first step and required for the beta oxidation. So as you can see that the acyl coenzyme A synthase, which is also called as a thiokinase, it is embedded in the outer aspect of the outer membrane, and it results in the formation of this acyl coenzyme A. This acyl coenzyme A finally get complex with the carnitine molecule, and this reaction is catalyzed by the enzyme known as the CPT1. This C represents the carnitine molecule. This CPT stands for the carnitine palmitoyl translocase 1 enzyme. It is embedded in the inner aspect of the outer membrane. As you can see, this is the outer membrane. It results in the formation of a molecule known as acyl carnitine. This carnitine molecule helps in the translocating the acyl coenzyme A molecule from the outer membrane to the mitochondrial matrix, where the machinery required for the beta oxidation is present. So once acyl carnitine is formed, there comes another enzyme known as the carnitine acyl carnitine translocase enzyme. It is embedded in the outer aspect of the inner membrane. You can assume that from its right hand, it transfers the acyl carnitine molecule from the outer membrane to the mitochondrial matrix, and from its left hand, it transfers back the simple carnitine from the matrix to the outer membrane. So in this way it works. You can assume it as a counter transport mechanism. Well, there comes another enzyme embedded in the inner aspect of the inner membrane known as CPT2, the carnitine palmitoyl translocase 2 enzyme. It cleaves the acyl molecule from the carnitine, and this is that carnitine that is translocated back to the outer membrane from the mitochondrial matrix by the carnitine acyl carnitine translocase enzyme. Anyhow, this acyl molecule is then connected again to this coenzyme, finally forming the acyl coenzyme A back again in the mitochondrial matrix, and this is the acyl coenzyme A which undergo the beta oxidation processing. The carnitine molecule itself, here its chemical formula, it is beta hydroxy gamma trimethyl ammonium butyrate. It is chiefly synthesized in the liver by the lysine and methionine amino acids. One thing very important about the carnitine molecule, that it is very important for the long chain fatty acids, as the shorter chain fatty acid can enter the mitochondria and they can undergo beta oxidation spontaneously. So the carnitine molecule is especially important for long chain fatty acids, as the mitochondrial membranes are impermeable to the longer chain fatty acids. So once the acyl coenzyme enters the mitochondrial matrix, it has to face the fatty acid oxidase complex. It's a complex comprising of several enzymes like acyl coenzyme dehydrogenase, enoyl coenzyme hydratase, and beta hydroxyacyl dehydrogenase. There is one more enzyme known as thyrase enzyme. It's also part of the fatty acid oxidase complex. Anyhow, the first step that happens in the mitochondrial matrix is the dehydrogenation. The hydrogens from the alpha carbon and one hydrogen from the beta carbon, they are removed by the enzyme acyl coenzyme dehydrogenase, and the product is the alpha beta unsaturated acyl coenzyme A. The flavor protein containing the FAD as its phosphatic group is reduced, which is used for ATP synthesis in the electron transport chain. This reaction is catalyzed by the acyl coenzyme dehydrogenase, and like the acyl coenzyme synthetase, the acyl coenzyme dehydrogenase has several types, each specific for a specific length of the fatty acid. The next is the hydration reaction. The alpha beta unsaturated acyl coenzyme A is also called as a delta square trans-enoyl coenzyme A. It gets hydrated in the presence of enzyme enoyl coenzyme hydratase, which is also called as a crotonase, and the hydroxyl group of the water get attached to the beta carbon, while the hydrogen of the water get attached to the alpha carbon. So it results in a formation of the beta hydroxy acyl coenzyme A. The next reaction again is the dehydrogenation, in which the beta hydroxy acyl coenzyme A, the hydrogen of the hydroxyl group attached to the beta carbon, and the hydrogen which is attached directly to the beta carbon gets removed. So the resultant compound is the formation of a keto group over here on the beta carbon, which is the beta keto acyl coenzyme A, which is catalyzed by the enzyme beta hydroxy acyl coenzyme dehydrogenase. I highlighted these two because these NADH2 and FADH2, they are important in the energy production, as these reduced coenzymes are reoxidized in the electron transport chain, and they are utilized for the energy production. So the final step of the mitochondrial beta oxidation is a thiolytic cleavage. It involves the splitting of the bond between the alpha and previously known beta carbon, which is now a keto carbon. So this reaction involves an enzyme known as thiolase, and the coenzyme A is also inserted to get attached to over here. So the compound can be converted into the acyl coenzyme A and acetyl coenzyme A. This acyl coenzyme A would have two carbon less as compared to that acyl coenzyme that had entered into the mitochondrial matrix initially. Once this acyl coenzyme had been formed, it undergoes dehydrogenation, hydration, dehydrogenation, and thiolytic cleavage, and the cycle continues until the whole carbon chain is converted into acetyl groups. And in this way, the whole fatty acid is degraded, and this is called oxidation, as it attacks the beta carbon, and the beta carbon takes the position of the functional group. So that's why it is called as a beta oxidation. Now this is was something about the carbon chain having even carbon atoms. If a carbon chain had odd number of carbon atoms, so all the cycles would be the same, but the last step would be that alongside the acetyl coenzyme A, there would be an palmitoyl coenzyme, which is 3-carbon compound, and that palmitoyl coenzyme A would be the only glucogenic part of the fatty acid. It would be converted into the succinyl coenzyme A, and this succinyl coenzyme would enter into the Krebs cycle and would produce energy. So this was something about mitochondrial beta oxidation. Now there is a unique type of beta oxidation known as a peroxisomal beta oxidation. The substrate of the for the peroxisomal beta oxidation are very long chain fatty acids, as their enzyme do not attack on the short chain fatty acids. The unique feature of this peroxisomal beta oxidation is that its products are acetyl coenzyme A and the hydrogen peroxide. In the mitochondrial beta oxidation, what happens that when the dehydrogenation is occurring, there are oxidized coenzymes available which are converted to reduced form. But in case of peroxisomal beta oxidation, there are no such coenzymes available. So therefore, the hydrogens which are removed during the dehydrogenation step are used in the formation of hydrogen peroxide. So as the hydrogen peroxide can't produce energy by going through the electron transport chain, so therefore one unique feature of the peroxisomal beta oxidation is that it produces no ATP. And the enzymes of the peroxisomal beta oxidation are induced by high fat diet. So this was all about beta oxidation. We have talked about the mitochondrial peroxisomal beta oxidation.